For exercise 4, we're going to be covering the cell. The goal of the cell is to speak to more of a generalized structure, organization, function of a cell. We're going to start with what is called a composite cell, and then we're going to, as we go later throughout the semester, start speaking to specific cells in specific organs and specific organ systems. But right now we're going to start with just a general cell. So what is a cell? A cell is the structural and the functional unit of all living things. So it is the foundational piece that needs to be there in order to build and have anything work functionally. So you'll notice it says all living things. There is cell biology, plant biology, animal biology. All of those come into play. We are going to specifically start speaking to, at this point forward, cells that relate to the human living things, not so much all of the other areas of science. So some specifics about cells that are highly important to this whole concept. They are very, very diverse. So there are lots of different types of cells. We're going to talk about probably close to 30 different types of cells this semester, and you'll probably double that number or triple that number by the end of AMP2. So there are a lot of different types. It is very, very, very diverse. They are actually different in their size and their shape. And if they are different in their size and in their shape, what we know to be true in AMP is that means that they are eventually going to be different in their function, since their structure determines how it actually works. And if they have different size and different shape and different function, they are all going to have very specific roles within the human body that they are going to need to be able to accomplish and kind of own in order for the overall system to work. So, what are some common things of our cells? We're going to start with talking about something that is called a composite cell. So this is going to be the anatomy of a composite cell, is what we're going to talk about next. Composite cell is going to be a general overview for a generalized cell. So we are simply taking all of the features that are typical or normal in a cell, and we're going to talk about the anatomy of all of those. As we go throughout this course, some of the cell types we'll talk about will not have some of these features. Some cells will have these features plus a whole lot of extra things. So this composite cell is going to be a general perspective of all of the cells kind of combined into one big overarching overview. So there are 3 major regions to a cell, and we're going to break these down one by one. And with these cells, what we're talking about is these are the cells that we're able to identify by a light microscope, or the microscopes that we have in lab. The first major region of a cell is going to be the nucleus. So when we say the nucleus of a cell, essentially what we are saying is going to be the DNA and how you are actually going to create and organize and control the cell. So the DNA will be found in the nucleus. This is going to be your genetic material, and it is the instructions for how the cell actually needs to be created. You can also call the the nucleus the control center. It is responsible for making sure that the cell does what it needs to do and has all of the necessary tools to be able to use it. But what is really important for the nucleus is that this is going to be necessary for any type of cellular reproduction. So as we speak to the ability to have cellular reproduction, essentially what we mean by this is you cannot replicate the cell and make more cells without DNA. So in order to have more cells, you have to have the DNA. You only have the DNA if you have this nucleus. So what we'll talk about is you'll hear a term that is going to be anucleated. We'll talk about this a lot this course. If you ever see the A prefix, it means not. So if it is anucleated, it will mean not nucleated. And for cells that are going to be anucleated or not nucleated, there is no nucleus, there is no DNA. Those cells do not have the ability to divide. They essentially are created and then they die. They don't have the ability to divide. So we'll talk about that as we go throughout this semester. Inside your nucleus, this DNA will actually be found in two forms. One of the forms you're going to be probably highly familiar with, which is going to be a chromosome. You guys are probably familiar with humans have 23 chromosomes, but there's also another form of DNA that will be called chromatin. And we're going to talk through these specifically. So the DNA comes in two forms. Chromatin is where there is no division occurring. Your DNA is not getting ready to divide. The way we know that this DNA is in its chromatin form is the DNA is loose and dispersed. So that means what we are saying loose and dispersed, your DNA will look like this throughout the cell. It is loose and it is dispersed. But when we're talking about the DNA in the chromosome form, what we mean by that is it will be DNA in a form that is called coiled. And this coiled will be ready for reproduction. So it's the same information, it's the same function, it's the same essentially foundational piece, but they're going to be in two separate forms. Chromatin will be loose, chromosomes will be in their coiled form. So it's the same information, will just be in two different forms. So that's important for the nucleus. Another important part of the nucleus is going to be something called the nucleoli or nucleolus. You'll see those terms, nucleolus and nucleoli. This is where you will find specific proteins that are necessary for the reproduction of the DNA, and you will find your RNA, which is important for building new proteins. So this is basically going to be a place where you will find the information for protein synthesis, is these nucleolus. And you will be able to create new proteins. That will come highly important a little bit later in the course. And then the other structure that is involved with our DNA or our nucleus is going to be our nuclear envelope. So our nuclear envelope is essentially the outside border. So this is going to be the double layer membrane around the nucleus. So this is a double layer membrane and it will surround the entire nucleus. So if we were to draw this guy out, essentially your nuclear envelope looks like this, but not just like that, because it is going to be porous. Woo. And by porous, we means it will have nuclear pores. And if you think about the pores in your skin, they're essentially going to be spots on your skin, but they're also going to be holes within your skin. So when we're talking about something that's porous, your nuclear envelope will actually have nuclear pores that make it that are going to make it so things can travel inside and outside of the nucleus. Okay, so this is our nuclear envelope. This is your nucleolus. And then if we were going to have our DNA in the structure where it is going to be dividing, our DNA will be in chromosome form. So this is what the inside of your cell will look like. This is going to be the inside of the nucleus. You'll have your nuclear envelope, your nucleolus, and your DNA or your chromosomes form. When you're looking at this underneath a microscope, essentially the nucleolus will actually look like a marble. The nuclear envelope kind of looks like a cup, and then this whole thing is going to be inside the actual cell. And that's our next thing that we're going to talk about, is going to be the plasma membrane. So the first part of our cell was going to be the nucleus. The second major region of our cell is going to be the plasma membrane. Plasma membrane. So when we're looking at the plasma membrane, what we're looking at here is the structure that will surround the cell. It's the fence. It's the wall that will keep everything inside the cell and then keep the things outside the cell that need to stay outside. So it will surround the cell and it will separate it from the environment. It will maintain a boundary, which is very important for maintaining the integrity of both the inside and the outside. So this plasma membrane is really, really, really important. The things that will make up your plasma membrane, the building blocks, it is going to be made up of fats called phospholipids. It will be made up of proteins, and it will be made up of carbohydrates or sugar chains. So these are the three primary ingredients that make up your plasma membrane, but the phospholipid, the fats, is going to be the priority. So one thing that is super interesting about this plasma membrane is, here we go. Here is our plasma membrane around our cell. We've already talked about this is going to be our nucleus. Remember, it's got pores. Here's our nucleolus, and here's our DNA in its chromosome form. So what we're talking about is this outside wall, this plasma membrane. So one thing that is super important about this is that it will provide protection for the cell because it acts as a barrier. So it will both protect things that should not come inside, and it will protect things that should stay outside. It is selective, meaning it does act as a wall, but it also has the ability to let things into the cell that need to come into the cell, or let things out of the cell that needs to come out of the cell. So it's selective to transport. Some things can be transported in, some things can be transported out. And how it is transported is going to be whether or not the plasma membrane likes it or doesn't like it. So it's going to be selective to those features. It will allow in nutrients. That's going to be the primary reason things will need to come into the cell. Nutrients will need to come in. And then it will need to actually keep out or kick out harmful substances and waste. So these are the things that we will want to remove from the cell. Harmful substances need to keep out, and waste needs to be removed. Because of these features, your plasma membrane has a feature that is called selective permeability. And so this feature of selective permeability means that some things are allowed in, other things are not, but your cell actually is selective to the things that it will allow to come in. And the way it does this is because your cell has a very unique feature that is called a phospholipid bilayer. So lecture will go into more specifics about this phospholipid bilayer, but what you need to know is if I were to take this section right here, and we're just going to blow him up. This is the cell wall. So I'm going to blow him up. And essentially what you get is you have this. He is a bilayer, which means two layer. So if this is your cell, essentially it is going to be all the way around it. It is a two-layer system. It is a bilayer. And these bilayer cells are going to be made up of fat molecules, phospholipids, that look like this, that are essentially going to create two layers of this barrier. So here's layer 1 and layer 2. So these guys are going to make up the external layer. So your cell membrane or your plasma membrane is also called a phospholipid bilayer. It is two layers of phospholipids. And that is what allows it to have this selective permeability process. And it is very, very cool. Okay, so we had the nucleus, and then we had the plasma membrane, and there is a third feature to your cell that we need to talk about, and that will be the cytoplasm. The cytoplasm is essentially everything else. So this is going to be the major site of cell activity. So this is where you will find the majority of normal cell functions is going to be in the cytoplasm. But the cytoplasm will be responsible or be made up of two things when we're talking about the cytoplasm. We are both talking about the cytosol, which is going to be the fluid inside the cell. And we are also talking about organelles that are in the cytosol. And these are going to be small structures. So remember, organs are going to be part of organ systems. So your organ could be your heart, your liver, your lungs. Organelles are going to be small, teeny tiny little structures that operate that same way, where they are responsible for a specific function. So inside your cytoplasm, you'll have the liquid, which is the cytosol, and then you'll have your organelles. We're going to talk through these organelles specifically. There's a bunch of different ones, and we're going to talk through those. But to give a frame of reference, here is our plasma membrane. Here is our nucleus. Here is our nucleolus. And here is our chromatin. So your cytosol is going to be everything that is liquid inside. So your cells are not actually empty. They are full of liquid. So your cytosol, these will be full of liquid. And then we're going to talk about a bunch of different type of organelles, structures that will live inside your cytoplasm. And this is where you will have the functions of the cell occur. So what are these organelles, and what are we going to talk about? So we're going to hit the highlights of these organelles for lab. The organelles that are found inside your cytoplasm. The first one is going to be called ribosomes. So when we're going to talk about these organelles, we're first going to name the organelle. Then we're going to draw the structure. What does it look like? So we're going to just draw that out. And then we're going to talk about the function. What does it actually do? Because remember, in human anatomy and physiology, the structure determines the function. So what is it called? What does it look like? And then what does it actually do? So that's what we're going to talk about for these organelles. So the first one is going to be ribosomes. I'm just going to make it smaller. So the structure of ribosomes are going to be this. And that is it. They are small spherical bodies. They are very small little balls. They are spherical bodies, and they are going to be made up of RNA and protein. So they look like little tiny dots. The function of ribosomes is going to be protein synthesis. Synthesis means creation of or development of. So when we say protein synthesis, we mean that these guys are responsible for creating new proteins. So ribosomes are responsible for creating new proteins for protein synthesis. The next organelle we're going to talk about is going to be the endoplasmic reticulum. Endoplasmic reticulum, otherwise known as the ER. So there are two different types of the endoplasmic reticulum. You will have the smooth ER, and you will have the rough ER. Smooth ER will have a structure that is different than the rough ER, and if they have a different structure, therefore they are going to have a different function. So remember those pieces. So the ER in general is going to be a very folded systems of tubes. So when you're looking at the endoplasmic reticulum, it is a folded system of tubes. So it will actually look like, I like to call this guy a little bit more like ramen. It is a series of tubes. And with these endoplasmic reticulums and these series of tubes, if it is smooth, that is all it is. If it is rough, it is going to be that series of tubes that kind of looks like ramen. And then what it will actually have is it will have bumps all the way around it. It's going to be rough. The way I like to describe this is if you've ever had nerds, nerds are going to be these ribosomes. I'm talking about the candy nerds, the little tiny, little round things that you bite into and it's sugary. The candy nerds are the ribosomes. The endoplasmic reticulum is actually going to be licorice. It's smooth. There's nothing attached to it. and it's just a bunch of tubes that are put together, whereas your rough ER is going to be the nerds on a rope, where it is going to be your licorice that has all of these little ribosomes embedded into it to make it very, very rough. So your rough ER is actually going to be with ribosomes. So it is your ER that has ribosomes attached. So that's the structure. What is the function? The smooth ER is responsible for steroid and lipid synthesis. Steroid and lipid synthesis. So steroids and fat synthesis. Whereas your rough ER that has your ribosomes is actually going to be storage and transport of proteins. So I'm going to stop right here and talk through the difference between these guys. So remember, up here in our ribosomes are these small little spherical bodies, and that's where you create new proteins. It is protein synthesis. Well, down here in rough ER, which is going to be all the tubules that has the ribosomes embedded into it to make it rough and bumpy, this is where you store and then transport those proteins. So remember, the ribosomes are related to proteins. The smooth ER, which is just a bunch of tubules that does not have ribosomes attached to it, has nothing to do with proteins. And it is going to be steroid and lipid synthesis, or steroid and fat synthesis. So that kind of helps keep all of those guys separate. Another structure is your Golgi apparatus. And your Golgi apparatus looks like flat stacks of pancakes that are all on top of each other. So this is your Golgi apparatus, flat stacks of pancakes. And this is where you will actually package proteins. And it's where you will package waste. And the reason you are packaging these is because they need to be exported out of the cell. So the way I like to describe the Golgi apparatus is this is the FedEx. He's the guy who will put the things that need to leave the cell or need to be mailed somewhere into a little package, into a little box, so that they can be moved from cell to cell or out of the cell. So the Golgi apparatus is responsible for packaging whatever needs to leave, so that way it can be exported from the cell. Another structure inside the cell, another organelle, is going to be lysosomes. So lysosomes are going to be different size little fluid-filled sacs. So these are fluid-filled sacs that are going to be different sizes. So what is inside the lysosome is the important. What the fluid is very important. So inside the cell is going to be something called a hydrolase. A hydrolase liquid inside the cell is actually going to contain digestive enzymes. Enzymes are something that's going to make the reaction occur. So an enzyme that's a digestive enzyme is going to make the digestion occur. So these little fluid-filled sacs have a liquid inside that causes digestion to occur. Well, if you are inside the cell, these lysosome sacs, when they break open, they are actually called the suicide cell or suicide sac of the cell. Because if these guys are broken inside the cell, they will actually start to digest the cell. Because they are containing the enzymes that are responsible for digesting things, and they should be digesting other things, but if they are broken, a lysosomes will release its hydrolase or its digestive enzyme. It's called the suicide sac of the cell, because it will essentially start to digest the cell from the inside out, which will destroy the cell. Another thing that you have inside as an organelle is going to be peroxisomes. Peroxisomes are also small fluid-filled sacs. They look almost identical to the lysosomes, but they are different. Their fluid that they are containing is not a hydrolase. They will actually contain an oxidase liquid. So they will contain an oxidase liquid inside. And this oxidase enzyme essentially is a detox enzyme. And it will pull harmful chemicals out of the body. So it will detox harmful chemicals from the body. Specifically, it's responsible for helping out with the liver detox and alcohol. So your peroxisomes or oxidizes, they will actually pull out and detox harmful chemicals, which is awesome. Okay, you're going to have two more structures inside the cell that we're going to talk about. You're going to have mitochondria, which I'm sure everyone has heard about and has heard it called the powerhouse of the cell. So the mitochondria actually looks like a little oval structure, oblong like this, and it is going to be a bilayer as well. And it will be a bilayer with folds, or intramembranous folds is what it's called. So when you have a bilayer with intramembranous folds, that means that it will look like there's a fold, 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 there's a fold. So this is what it will actually look like, where there is a bilayer. There's one layer all the way around, and then the inside layer, the membrane, is going to be folded. So mitochondria, you've probably heard this called the powerhouse of the cell. It is very, very, very true. That is what it is. But on a actual scientific level, and kind of building further on to the powerhouse of the cell, is these mitochondria are going to contain enzymes. And they are going to contain the enzymes that are responsible for breaking down foodstuff. And after they break down foodstuffs, they are going to convert that to what is called ATP. So mitochondria at the end of the day help break down things because they only care about really producing ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate. So part of AMP, you should know that ATP is adenosine triphosphate. Mitochondria produces ATP. ATP is the gas of the cell. It's the chemical that makes us go. It makes us actually be able to do any type of energy. It's the electricity. So mitochondria is really important, and it's where you produce that energy, that ATP. The last thing we're going to talk about in your cell is going to be centrioles. And centrioles look to me like pieces of sticks that are stuck together, and then what they'll have is that they're going to be at right angles to each other. So it kind of looks like a bunch of sticks or a bunch of pencils, and they usually are found at right angles. So they're usually found with one set like this and one set like this. And these are actually going to be really responsible and important during cell division, which we're going to talk about next briefly. So cell division is where you actually produce new cells. You go from 1 to 2 cells. So centrioles are very, very important to this process. Okay, so what we're going to talk about next is that's the general components of the composite cell, the 3 main regions, the nucleus, the plasma membrane, and the cytosol, and then all of the organelles. Next, what we're going to talk about is going to be the cell cycle. So when we're talking about a cell cycle, we are talking about all the changes that a cell goes through, all of the changes that a cell goes through from formation to reproduction. So it's all the changes a cell goes through from formation to reproduction. You'll notice this does not say from birth to death, because cells do not work that way. They are not technically born, and they may die, but some cells don't actually go through a death process. From formation to reproduction. And the main reason is because there are 2 phases to this cell cycle. Phase 1 is called interphase. An interphase is going to be the long period of normal cell function. So this is you are just doing life. Nothing is happening. You are doing everything that you need to do as a cell in order to do the functions of the overall body. So interphase is just normal everyday life. And then it will go through a phase that is going to be called cell division. And cell division will actually divide the cell, just like it says. So when we say the cell cycle is changes a cell goes from formation to reproduction, and these are our two phases, what we mean is that you are going to have one cell. That one cell is going to be doing what is called interphase. That cell will then go through a process that is cell division, and he will produce two cells. These two cells will do their normal everyday interphase, and then they will produce two cells. So you'll notice that these cells were not born, they were produced, they were formed, and then these cells right here did not die, they split to create new ones. So this is not a cell cycle of life or death, it's a cell cycle from formation to reproduction. So we're going to spend time talking about the phases of cell division. This is what we're going to talk about next, is the phases that make up cell division. That is where we're going to live. So remember, this is not everyday normal life of a cell doing its function. Cell division is actually dividing the cell. It is making two cells. So cell division will actually follow two steps. Well, multiple steps, but two parts. The first part is going to be called mitosis. And mitosis is going to be the division of copied DNA. And these will divide from the mother cell, is what it's called, to the two daughter cells. So you are copying the DNA, you've already copied the DNA, and you're going to divide the DNA from one cell to two daughter cells. Up here, this is the mother cell, one cell, and then these are the two daughter cells. So we've essentially copied the DNA through mitosis. We've copied the DNA, and in mitosis, we're dividing the DNA. So the first step to mitosis is going to be called prophase. Actually, I'm going to do it a different way. So I'm going to do it this way. Prophase is the first step in mitosis. And prophase is actually going to have an early and a late phase, depending on what it needs to do. So in prophase, you're going to have early prophase, and you're going to have late prophase. But both of these are under the guise of prophase. So what's happening in prophase is your chromosome will actually start condensing, or your chromatin coils. If my chromatin starts coiling, I'm going to be in my chromosome form. So each chromosome will have two sister, what are called chromatids. So these will have two chromatids. So it's the chromosome coil that is put together. So we're going to have, here's my chromosome coil, and it will be attached at the middle. So chromosome coil, chromosome coil. So each one of these is a chromatid. So they're just attached in the middle. And then what's happening here is that your you will start to disappear in this prophase phase, where your nuclear envelope will start to break up. So your nuclear envelope starts to disappear. And as that nuclear envelope starts to disappear, what you will see is, I'm going to draw it right here. So here's the outside of my cell, my plasma membrane. Here, I am only going to draw the nucleus. I'm not going to draw right now the actual rest of the cell structures, all the organelles on the cytosol. So now we've got our nuclear envelope, and he is going to start to disappear. And inside, I will have my chromatids. put together where they are going to be attached by my chromatids, my sister chromatids. So it'll look like that. You'll also start to see your centrioles in the cell. These will be more clearly. Remember, these centrioles are going to be at 90 degrees from each other. So this is what you'll see with prophase. Then early and late. Early, you'll have a more clear nuclear envelope, and then by late prophase, your nuclear envelope will have disappeared. That's what will happen in prophase. The next phase after prophase is going to be metaphase. Metaphase only has one phase, so there's not an early or a late. So with metaphase, what is happening is you are going to have no nuclear envelope. So your nuclear envelope has disappeared completely. What you'll also have is that you will have microtubules from your centrioles will connect to the center of your chromatid. So your centrioles will connect to the center of the chromatid. And then what you will have is you will have all chromosomes lined up at the middle. So if this is the outside of my cell, remember I'm not doing any of the other structures. What we are essentially saying is I am going to have my microtubules. Here's one. Here's the other. Actually, let me blow that up a little bit more. And here are my sister chromatids. So just as an FYI, this is my chromosomes put together. I have two chromatids. I'm going to just draw them like this for ease of everything. But these are essentially the coils. But I'm just going to draw them separately, just a little bit easier. So what these guys have done is you have essentially lined up all of your sister chromatids down the middle between the two centrioles. And they have sent out these little tubes that are called microtubules, and these attach to the center of the centriole. So you'll notice that this one right here is going to send a microtubule to the center of this guy. Whereas this guy right here is going to send his to this side of the sister chromatid. So you are going to have, with metaphase, all of your nuclear material, all of your DNA through your chromosomes will be lined up perfectly down the middle. You'll have these microtubules that are going to be connected to the centrioles, and the centrioles are going to be at opposite polar ends of the area. And what's essentially going to happen is you're going to start pulling. And what you're going to do is the moment that you start pulling, that these microtubules, these guys right here, are going to start pulling. The moment that happens, you enter the next phase called anaphase. And anaphase is where you are actually pulling the chromosomes apart. And you are separating the sister chromatids. And as you are doing this, your chromosomes will uncoil into your actual chromatid form. So once this happens and you start pulling apart, your chromosomes are pulled apart, your chromosomes will actually do the uncoiling thing. So here's what it looks like. Here's my cell, my plasma membrane. Here's my centrioles. Here's my centriole. Up here, all of these guys on this side are going to be pulled in the direction of the green microtubules. So what they will do is these green microtubules will be pulling, and they will pull the sister chromatid, so they start doing this. Whereas this other side over here, these guys, these microtubules, are going to be pulling these guys. So they will be pulled and they will start looking like this. And you'll notice when this happens that there's going to be a wide space between them. So they'll start to pull apart. So what is actually happening with this guy? What I like to do is I like to describe an umbrella. If you are outside in the rain, this is what your umbrella looks like. But what happens if it's really windy? Your umbrella goes from this to essentially inverted like this, right? If it's really, really windy, your umbrella actually blows up. Well, that's what's happening with these sister chromatids or these chromosomes. They essentially are going to be like this. They're going to be this umbrella, this one right here. But the moment that you start pulling on this middle spot, this one right here is going to pull this way. And this one right here is going to pull this way. And what you're going to do is you are going to pull the center out. You're going to invert it. So you're going to essentially do this. You are flipping your umbrella upside down. And the same thing's going to happen. If I take this guy and I pull from that center point, he's going to flip. And when he flips, I've essentially inverted my shape. So it's kind of like an inverted umbrella. That's what's happening here. With anaphase, you are going to have the space and you are going to pull these all the way to the sides of the cell. So you will actually split the DNA into two parts and they will travel to different sides of the cell. Okay, the last phase of mitosis is called telophase. And with telophase, what you're actually going to do is this is going to be the division of the plasma membrane. So everything else that we've talked about has been the DNA so far, but telophase will be the division of the plasma membrane. So you'll notice right here, I drew a circle, but the moment that you get a little bud right here and a little bud right here, you'll start to see, here's this, and then you'll pinch it off essentially in the middle right here. As you're pinching it off in the middle, you'll have one centriole over here, one centriole over here, and then you will have the set of DNA over here and the set of DNA over here. And if you pinch off this middle section, you are essentially going to keep going until you have created a division between the two. So telophase is where you are actually budding off the middle and you are going to be creating the two different daughter cells. What's also happening in this telophase is you will essentially reform the nuclear envelope. And as you reform the nuclear envelope, you will start to put the surrounding layer, that bilayer nuclear envelope, back around the cell. At the end of telophase, when this is all said and done, you should have two daughter cells that have a centriole in each, a nuclear envelope, and the DNA. All of which has happened. Oh, as well as the nucleolus. All of which has happened because you just divided the DNA. So that's telophase. You're starting to divide the plasma membrane. During this entire process, I said that this was going to be the process of mitosis, and mitosis was the division of the DNA. So going from the mother cell to two daughter cells. So there's two parts to this cell cycle. We had, well, two parts to this cell division. We had mitosis, which was dividing the actual DNA, so that's these process. And then the second process is going to be called cytokinesis. And during this entire thing, we have split both the DNA and we have split the plasma membrane, but we have not split the third part of our composite cell that we talked about earlier. So cytokinesis is going to be the division of the cytoplasm. So it's the division of all the other stuff. And so the division of the cytoplasm will also include all of the organelles. So if our cells split into two, you need to have enough mitochondria go to one side that mirrors the same amount of mitochondria that go to the other. You need to have the Golgi apparatus split in half. Not necessarily the actual structure split in half, but if you've got two of them, one needs to go to one side and one needs to go to the other to even it out. So cytokinesis is going to be the division of the cytoplasm, the liquid, and all of the other organelles in order to maintain the integrity of all of the structures inside the cell. This will begin during anaphase, and it will go through telophase. So up here, when we're talking about mitosis, while anaphase and telophase are occurring, while these guys are occurring, you are also going through cytokinesis, where you are dividing the cytoplasm and you are also dividing the organelles of the cell. Okay, so that's the cell cycle and what you need to know about this. For the lab this week, what you guys need to focusing on inside your lab manual. So again, we are at exercise 4, the cell. And so you need to look at activity 1. And activity 1 is going to be, it would be a whole lot easier if you were in the lab, but you can do this on your own. So it's identify the parts of the cell. So everything that we talked about at the beginning, the composite cell, the cell membrane, the cytosol, the organelles, and the nucleus, please make sure you can identify the parts of the cell. You also need to do activity 2. Activity 2 is going to be all the parts of the plasma membrane. So see if you can identify what the lipids were, what these other pieces that make up the plasma membrane. Activity 3 is going to be all of the organelles. So with these, you do need to know both the structure, what they look like, and you need to know the function. So if you were to see a picture of the actual organ or organelle, then you could tell me what the actual function of that is. So you need to be able to do the structure and the function and find the organelles inside the cell. And then the last thing that we are doing is going to be activity 4. Well, I guess activity 4 is will be really helpful through using mastering AMP. So activity 4 is looking at the cell model, finding all of these things in a global picture model of the cell. There's one in your, I think, reading quiz for this week that is super helpful. If not, there's one in the back of the book. And then also I'm going to add that you guys look at activity 6, which are the mitotic stages. And the cell that I would like you guys to draw is look at underneath PhysioX, the mastering AMP. I want you to draw mitosis, but not all of the phases. I want you to draw the mitotic stage of metaphase. I want you to draw the mitotic phase, metaphase. And that is your histology drawing for this week, is metaphase. And that is it for exercise 4.